Hi everyone, my name is Laura and the title of my research project is Investigating the Association Between Prenatal Stress and Neurodevelopment, the Contribution of the Gut Microbiota. To begin, approximately 30% of pregnant individuals worldwide report significant stress during pregnancy. Prenatal stress is associated with adverse neurodevelopmental outcomes in children. For this reason, researchers in the field of maternal and child health want to understand how prenatal stress is transferred from the mother to the baby, and how stress can program the fetal brain. Emerging evidence suggests that the gut microbiota may mediate the relationship between prenatal stress and neurodevelopment. The gut microbiota refers to all the bacteria present within the gastrointestinal tract. Children are first colonized by bacteria during the birthing process, where bacteria are passed on from the mother to the child. Bacteria within the gut are essential to human health because they can produce metabolites like short-chain fatty acids and neurotransmitters, which can be sensed by the central nervous system. Both human and animal studies have linked the gut microbiota to cognitive and mental health outcomes. Interestingly, prenatal stress is associated with altering the child's gut microbiota composition. However, very few studies have examined the relationship between prenatal stress, gut microbiota, and neurodevelopment. For this reason, the objectives of the study were to first determine if the amount of prenatal stress exposure is related to the composition of the child's gut microbiota in children aged 3 to 4, and second, determine if the children's full-scale intelligence quotient and primary indices scores are different depending on the composition of the child's gut microbiota. I used data collected from the Alberta Pregnancy Outcomes and Nutrition Study to answer both objectives. I operationalized prenatal stress using both a biological and psychological measure at each trimester separately. For the biological measure, I used maternal salivary cortisol samples, and for the psychological measure, I used two mood questionnaires, the Ettenberg Postnatal Depression Scale and the Anxiety Subtest of the Symptom Checklist 90 to generate a psychological distress score at each trimester. Child stool samples were collected at the age of 3 to 4 and were used to determine features of the child's gut microbiota composition using 16S rRNA sequencing. Lastly, the Wechsler Preschool and Primary Scale of Intelligence, the WIPSI-4, was used for neurocognitive assessments. I would now like to draw your attention to the results section, specifically Figure 1, which depicts a correlation heat map examining the relationship between the maternal stress measures in each trimester and child alpha diversity measures. Alpha diversity is an index that lets us know the number of different bacterial species found within the gut. In other words, this index is a macroscopic index that allows us to understand how diverse the gut microbiota composition is. The blue indicates a positive association between variables, and the red indicates a negative association. On the left side of the graph, we see that maternal cortisol in trimester 1 has a significant positive association with alpha diversity. In contrast, maternal cortisol has a significant negative association with alpha diversity in trimester 2. In comparison, if we look at the right side of the graph, we see that the psychological distress score in trimester 2 has a significant positive association with alpha diversity. These results indicate trimester-dependent effects and suggest that maternal cortisol and psychological distress relate differently to the child's alpha diversity measures. I would now like to draw your attention to Figure 2, which is a correlation heat map examining the relationship between maternal cortisol measures and taxa abundance at each trimester. It was found that maternal cortisol exposure is significantly associated with 10 bacterial taxa in trimester 2. Lastly, I would like to draw your attention to Figure 3, which is a correlation heat map for the relationship between taxa abundance and full-scale IQ. There was a significant negative association between Shannon alpha diversity and the verbal composite score. To summarize, maternal cortisol exposure and psychological distress had distinct association with children's gut microbiota composition, and all these associations were trimester specific. However, compositional differences were not associated with child's full-scale IQ. Therefore, the current study findings indicate that child's gut microbiota does not mediate the relationship between prenatal stress and full-scale IQ in children aged 3 to 4. However, these findings are important because previous research suggests that differences in microbiota composition are associated with differences in cognitive outcomes in early childhood.